Okay, guys, before we launch into the different kinds of plasmids, I just wanted to mention, now that we've gone over transformation and conjugation, that not all organisms, not all species, take part in all of these different types of horizontal gene transfer. For example, conjugation is quite common among gram-negatives, not as common on gram-positives. And the other thing I want to mention is that when you have a pilus form during conjugation, this can happen between two members of the same species, but it can also happen between members of different bacterial species. And this is pretty crazy when you think about it, because not only are these organisms going to be able to use those genes and express those genes real time in that same generation, and then also pass that down to the next generation vertically, but they're able to get these characteristics and these abilities from totally different species. You don't see this in the animal world, right? Really much at all. Even in vertical gene transfer, you'll see a couple of examples with a donkey and a horse, right? They make a mule, and that's not even it's a sterile individual. It's not able to reproduce. Or lions and tigers can mate. But that, one, is not horizontal gene transfer, and two, it doesn't allow for the genes to perpetuate because the offspring are sterile. So this is pretty crazy, powerful stuff. Let's see how much more powerful it is by talking about different plasmid types that exist. Okay, so the first one, and it's one that we've actually kind of touched upon, is a conjugative plasmid. Like a conjugal visit, think of it that way, right? A conjugal visit, and these are sometimes called F plasmids because these make the actual bacterium fertile. Right? So fertility plasmids is the way where they're thinking about it, okay? These will contain plasmids with an origin of transfer. So in other words, the plasmid contained within the bacterium is capable of spinning off new plasmids into the adjoining cell, okay? It's that origin of transfer, which is the location where there'll be a little bite taken out of one side of that double-stranded DNA, and a DNA polymerase will produce a new strand as it goes into the other cell. Also, these particular plasmids have the gene for the pilus. So not only would a cell receiving this plasmid become fertile, become itself a conjugative cell and have a conjugative plasmids, but it would then be able to make the pilus and pass the plasmid on to other cells. All right, so we have that F plasmid, fertility plasmid, or conjugative plasmid. We also see plasmids that are called resistance plasmids, and that's kind of a no-brainer if you think about what that probably is about antibiotic resistance, all right? R plasmids, or resistance plasmids, give a resistance and protection from antibiotics that exist out there. Now, one example is R100. R100 happens to be found in certain bacteria. Some examples would include E. coli, so Escherichia, that's the genus, the E in E. coli, Klebsiella, Proteus, Salmonella, the list goes on and on. Now, all of these are gram-negative. So I had a couple of questions earlier from people, asking if gram-negatives were more dangerous. And my answer was kind of both can be dangerous and cause some pretty nasty effects. But what you're thinking of when you think about gram-negatives is the ability or inability to kill them, right? If you have an infection. And these organisms, these enteric organisms, all these are found in the intestines. And many of them, are common normal flora that we live with in harmony. But if they find their way into places other than the intestines, if there's fecal contamination of something, then it can cause some real problems. Now, what does this R100 plasmid do that these organisms contain? It gives resistance to sulfa drugs. Okay, we're going to talk about these. Sulfonamides are a sulfa drug. Streptomycin, spectinomycin, fusidic acid, chloramphenicol, and tetracycline. Now, that's not just one antibiotic that these organisms would be resistant to, but several types of antibiotics, right? Now, let's add to the power here of what we're talking about. We know that sometimes plasmids will have similar sequences in their genetic code, and they could incorporate themselves into the bacterial genome. And then as that organism went through binary fission, it would just perpetuate that genetic material. It would just continue. Well, what if an F plasmid that contained the ability to produce a pilus and to transfer genes through conjugation through that cylinder, that pilus, what if it crossed over, recombined with a resistance plasmid? Well, now we have an organism that is going to be able to use this plasmid to actually pass through the pilus, not just genes for the pilus, but genes for antibiotic resistance. All right? And those antibiotic resistance genes can be used real time, right there and then, not having to wait for the next generation, though it can be passed to the next generation vertically. All right, some other plasmid types. We have bacteriocin or antibiotic plasmids. Now this is different. I'm gonna step back here to this other slide we just did. Resistance plasmids are containing genes that give resistance to the antibiotic. They don't produce the antibiotic. And you might say to me, well, are you nuts? We use the antibiotics. We're the one introducing the antibiotics. But I want you to realize that we are only stealing the idea of the chemical that causes death, the antibiotic. We're just stealing that idea from lots of other organisms that naturally make antibiotics, okay? It's a dog-eat-dog world out there. There's a lot of organisms in the soil, organisms in the ocean, organisms inside of us, and they are fighting for space, nutrients, all that. So antibiotics are produced by microbes to kill other microbes, okay? So that they can take the nutrients in that region, so that they can reside in that space, okay? So what's the difference between an antibiotic, a chemical that kills, and a bacteriocin, okay? Bacteriocins are chemicals that inhibit or kill close relatives, versus antibiotics inhibit or kill a wide variety of organisms. All right, so let me give you my analogy for this, and hopefully it'll help. So my wife and I used to shoot photography, and we still have a bunch of photography equipment, but for real, we used to shoot photography kind of part-time. We did weddings. I think we did over 50 weddings, but we don't do it much anymore. But so we shot over 50 weddings, and we knew exactly what other wedding photographers in the area, the Binghamton area and surrounding areas, charged. We knew what products they would provide their customers, what kind of albums they would give them, and access to images and things like that afterwards. So we knew those are like our close relatives, right? Those folks are just like us. Some of those smaller companies that would just do wedding photography. So we would want to, provide enough for a price that is right, that we would get some customers, right? And get some people that would hire us. Now, so we had a competition there, right? If I gave a little bit more and charged a little bit less than a close relative, then that will help me and it will hurt them, right? Now, let's think about as a different kind of idea, like someplace like Walmart, okay? And I don't hate Walmart, but we know Walmart has kind of a reputation for really ruining local businesses sometimes, because they do photography. They have photo labs, right? They may not be specializing in wedding photography, but they have photo labs. They've got gardening equipment, all stuff like that. So they're competing with True Value and Ace and all sorts of other companies that specialize in that kind of stuff. They've got grocery stores, right? So they're gonna compete with my little grocery store downtown in Whitney Point, right? So antibiotics kill a wider variety like that. They're going to affect a wider variety of organisms. So that is the difference. And we're saying that genes for the production of these chemicals exist on plasmids. And we're learning they could be shared between organisms. Okay, a fourth type of plasmid might be a physiological plasmid. So this would be a plasmid that has a certain physiological ability. Perhaps it produces fimbriae. It's a physiological capability they have, a gene for fimbriae, and so they're able to attach to things. They're able to attach to their host, maybe. They have a particular protein on their surface that allows them to utilize a sugar or to take in a sugar and use it for a nutrient source. These kinds of things, a physiological plasmid, can be really beneficial to a cell. A virulence plasmid. Okay, so an example here might be maybe a plasmid with a gene that codes for hemolysin, for an enzyme that breaks down red blood cells, like we've seen in our agar plates, right? So E. coli might make a certain amount of hemolysin, and this will break down red blood cells, and they get nutrients from that. Or enterotoxins. These can be coded for on virulence plasmids. And some of these actual genes give their host some real trouble, right? They're helping the organism to survive, perhaps, but the fact that they're virulent means that we're going to have all the signs and symptoms associated with the disease because it's so virulent. Okay? All right, so understanding now, I hope you see the gravity of this. Plasmids can be shared in real time. When a cell dies, and it doesn't have to be naked little linear pieces that spill out of a cell, it could be these plasmids. And then they're taken up through transformation. Even though they're circular, you can have transformation of circular pieces. Okay? Or these could be moving through conjugation from one cell to another. Right? So very powerful thing. And if you think of a disease, let's say an infection you have, a stomach bug or whatever, as long as make sure it's bacterial, right? If you have a bacterial infection, it's causing some kind of disease or some kind of infection. And you take antibiotics against that infection. Let's do a UTI, right? You have a urinary tract infection. And you start feeling better after a couple days. Well, in those 3 days, you probably killed a good number of the sensitive bacteria. Bacteria that do not contain, let's say, resistance plasmids. And you start feeling better. So you say, I'm not going to take this drug anymore. Well, guess what? There's a certain percentage of those organisms that may be resistant, and your immune system could handle those, like a small percent. Let's say there's 10% of that total infection. But if you don't take all of those antibiotics and you leave a good portion of the sensitive cells to now borrow, right? Take from the other resistant cells, those resistance plasmids, well, now you have a different monster, right? you may find that that antibiotic is no longer effective and your infection comes back. You'll have to switch to a different antibiotic. Okay, so understanding that this kind of sharing takes place really shows us the power these organisms have and explains to us how we need to act smarter when it comes to treating the infections that may have these kinds of plasmids. All right, let's call it quits, and we'll do the other two kinds of transformation. I'm sorry, other two kinds of horizontal transfer next.